Hey, this is Zagam from Z Biology. Today we are going to study about the structure of DNA. Starting from the cell, all cells in our body contain DNA. We have different kinds of cell in our body, each having its own function. But if we look at each cell, the main component of cells may be the plasma membrane, the nucleus, and the cytoplasm, of course. Additionally, each cell have many organelles like mitochondria, centrosomes, vacuoles, etc. Today we are going to focus on the nucleus because the DNA is present inside the nucleus. DNA present inside the nucleus is in the form of chromatin material, and it is only visible at the time of cell division. During the cell division, the DNA is visible in the form of chromosomes. This is one chromosome. This is a highly condensed form of a DNA. These chromosomes contain DNA wrapped upon special proteins called as histones. Let me draw a double helical structure of a DNA. This here is a double helical structure of a DNA. So, DNA is a polymer, and polymers are basically made up of monomers. The monomers of DNA are basically very small particles called as nucleotides. So the main game here is the nucleotides. If you understand the structure of a nucleotide, you will eventually understand the structure of the complete DNA. So let us look at the basic component of a DNA. A single DNA has a lot of these nucleotides. Now let us see what a nucleotide is. A nucleotide consists of three things. These three things are a sugar, a phosphate, and a nitrogenous base. Let's talk about sugar first. Sugar is present in the form of a furanose ring having 5 carbon atoms. The sugar is called as a deoxyribose. It's mainly a different form of ribose, just a missing OH group on carbon number 2. So that's why it is called as a deoxyribose sugar. Secondly, we will discuss phosphate that is present in all the nucleotides, a phosphate inorganic PO4 negative molecule. These two components are always the same in all the nucleotides. The component that changes in every nucleotide is the nitrogenous base. There are two types of nitrogenous bases, purines and pyrimidines. Purines are the double ring structures, while the pyrimidines are single ring structures. The double ring structures purines can be classified as two, like this one, which is called as adenine. And there is another purine that is called as guanine. Adenine is commonly referred as A, while guanine could be called as G, going towards pyrimidines. There are three different kinds of pyrimidines normally present in nature, called as thiamine, T, cytosine, referred to as C, and the third form of pyrimidine is the uracil, called as U. Now this uracil is not present in the DNA, that is only present in the RNA. The DNA has only 4 of these nitrogenous bases, that is A, G, C, and T. These bases join together to each other with hydrogen bonds, in which A is always bonded to T and G is always bonded to C. So, in a deoxyribonucleic acid, the monomer unit is a deoxynucleotide. Here I am drawing a deoxynucleotide. A deoxynucleotide contains this, a sugar unit, a phosphate joined at carbon number 5, and the nitrogenous base joined at carbon number 1. So this is a deoxynucleotide which has these three components. I hope it makes sense. So this was the basic structure of one nucleotide. There are many nucleotides in the DNA. Now for the joining of these molecules. These molecules join together with special bonds. Let me draw a nucleotide first. Here I have drawn a nucleotide and you can see there is the sugar. The carbon 3 I have drawn here will be used as the point of attachment for the next nucleotide. So this here is the structure of one strand of DNA, the single strand of DNA in which we have a sugar phosphate backbone. Similarly, the next nucleotide will join to this carbon 3 of this nucleotide. Now the attachment obviously is done with the help of special bonds. These bonds are called as phosphodiester linkages. Now let me draw the second strand of DNA. Keep in mind that this strand essentially runs in the opposite direction, hence drawn the upside down. But the sugar phosphate backbone here remains the same. The nitrogenous bases are the points of attachment from the previous strand. So here I am drawing the nitrogenous bases. You will see clearly that a purine has a pyrimidine in front of it. There cannot be a purine in front of another purine or a pyrimidine facing another pyrimidine. It has to be a purine bonded to a pyrimidine. They bind to each other with the help of hydrogen bonds. This here is complementary base pairing. This pairing is always done in a specific manner. The complementary base pairing is always from A to T and G to C. Adenine and thymine have double bonds, while guanine and cytosine always have a triple bond in between them. Hope you got the overall picture. Coming towards the direction of these strands. Now you can see that this strand here has carbon number 3 here and carbon number 5 is facing this direction. Now the opposite strand has 3 end at the top and 5' end at the bottom. We can say that this strand runs from 5' to 3' direction and this strand here is obviously from 5' to 3' in the opposite direction. Always remember, you will always mention the direction from 5' to 3'. Just the difference is both strands are antiparallel. They run in opposite direction. The structure here, the double helical structure, was first proposed by Watson and Crick back in 1935. So overall, we have two phosphate sugar backbones like this, running parallel to each other, joined together with the help of nitrogenous bases in between. In this double helical model of DNA, one turn is 3.4 nanometers long, having 10 nucleotides in it, and the total width of the molecule is 2 nanometers. The last point will be the grooves here. You can clearly see a smaller and a bigger groove. The smaller one is called as minor and the larger groove is called as the major groove. So that was the structure of DNA. If you have any questions, then please let me know in the comment sections below.